Right, hello, welcome to yet another revision stream. Why is there an echo today? There's a big echo. I'm gonna go close my curtains. There we go. So, basically, I've kind of just realised something pretty interesting about tomorrow's exam. So for those unaware, we were literally given the wrong exam paper. We were given tomorrow's exam paper yesterday. And I answered maybe 3 or 4 of the proper questions before we were given the right one. But during that time, it made me realise something. Either tomorrow, we're going to be given that paper again, which would be amazing because now we know exactly what's going to be on it. Or we're going to be given a different paper with easier subjects. Because what was on that paper was very difficult. There was some stuff on RNA, tRNA, protein synthesis, endopeptases appeared on there for some reason. A lot of stuff, which means it's a win-win. No matter what we revise, it should come up tomorrow, hopefully. So, I don't know what to do really, whether we should just go through the DNA topic or we should just start with the biological molecules. I'm honestly fine with doing whatever. If anyone's watching, feel free to comment. But anyways, I guess we'll just start off with the biological molecules topic. Now, I am I'm absolutely knackered after doing the math exam. So apologies if I miss out some stuff. I'm just going off what's off the top of my head. Okay. So she actually did specify what would be on the exam. She said carbohydrates. I'm gonna need a check. Hold on. No, I'll remember. She said transcription and translation, proteins, carbohydrates. So I guess if we start with carbs, this is probably the lengthiest topic. Keep doing a great job. Thank you, Matt. So, obviously, you got alpha and beta glucose. I always draw them the wrong way around. That's beta glucose. And the difference is that obviously alpha has the H on the top. Beta has the OH on the top. That's the difference. There are, obviously, you got the 3 main, you got glucose plus, okay, I always get these mixed up. I think it's glucose and fructose. Glucose and glucose and glucose and galactose. And that gives you lactose, maltose, and sucrose. Might have done that in the wrong order, but those are the monomers, those are the polymers. So basically, those are the disaccharides. Those are the mono. What else have we got for carbohydrates? We have, obviously, what do you call it? Starch. That's it. So starch is a storage molecule, meaning it's insoluble. Don't remember many other properties of starch, although I do know that it is comprised of 2 molecules. You got amylose, which is kind of like this, like kind of coils, and then you got amylopectin, which is like kind of a branched molecule. So obviously the branches allow this to be short and compact, or I guess just a better word, compact, which is great as a storage molecule. And amylopectin, obviously it's branched, which means that hydrolytic enzymes are able to more effectively hydrolyze the glucose for respiration. I saw one of the questions in the exam was about the structure of, was it is it? It's not glycogen. What is it? What's the other one? Shit. How have I forgotten? The other storage molecule, aside from cellulose. It's not glycogen. It's How have I forgotten the name? It was in the exam. I'm drawing a blank there, unfortunately. But I know starch is made up of alpha glucose. So is this molecule, which I've forgotten the name of. What I do know about it is long and branched. I think it's actually quite a small molecule, I believe, but it has a lot of branches. So it's an excellent storage molecule in animals. Or I guess you should say mammals, because I'm pretty sure that's what it's mainly in. So obviously the branches are for enzymes to easily bind. I forgot what you call it. It's not glucose. Glycogen. No. How have I forgotten? Glycogen, that's it. Great. I might have already said that, but it's just clicked now. That's it, glycogen. And then finally you got cellulose. So cellulose is made of beta glucose, which is something a couple of people forget. It's arranged in long unbranching chains, and it's linked via cross links of hydrogen bonds. And these form strong microfibers between the molecules. So hydrogen bonds, microfibers. I guess it provides rigidity for the cell wall, since it's the main thing in it. So that's carbohydrates. I'm going to get rid of all of this now, because I've taken up like half of the page. So that's all I can remember from carbohydrates. If you guys want to add anything, please pop it in the comments, because again, I'm on a tight schedule today. I have a lot of shit to go through, and unfortunately I'm only one person with a limited memory, so please comment what you can. I think I sent the link to Alex, but I have not sent it to anyone else, so complete RNG today. So we've done carbohydrates. Should we move on to transcription and translation and then jump back to all the other stuff? Yes, let's do that. So, transcription. Okay. It's very similar to DNA replication up to a certain point. So what we have is we have our DNA molecule. And I'm going to kind of draw it so that you can very clearly tell that there's two bases there. Hope that that's good enough for you guys up there. So you got DNA strand 1, DNA strand 2. Okay, so basically what happens is this unzips via DNA helicase. And the way it unzips is through the hydrogen bonds being broken between the bases. So the hydrogen bonds between the the nitrogenic bases are broken thanks to DNA helicase. So it breaks the H bonds. That's step 1. It's great. I've just gotten a message. Apologies for that. But just copy the definition. So DNA helicase, does it hydrolyze? I guess just breaks the hydrogen bonds here. So then from there you have the two separate strands, and what will happen is free floating RNA bases. I'll draw this in there. I don't have any other different colours. Okay, free floating RNA bases will join up to one strand. One strand. Okay? So they will join up to one strand of this now separated DNA molecule. And from there, you will get, I think it's RNA synthase. And what RNA synthase will do is it will not join the hydrogen bonds, because remember, we're not copying DNA here. Well, we're not like making a copy, like duplicating it, replicating it. Instead, RNA synthase joins these bases together by the phosphodiester bond. So RNA synthase joins the phosphodiester bonds together. Now from there, we now have obviously a RNA molecule, which remember RNA is just a smaller copy of DNA which is able to leave a nucleus. But before it can leave a nucleus, and I always forget this, it must go through splicing. DNA splicing. This is where the introns are removed and the extrons are kept. So let's just say that each one of these lines here represents a base. Let's just assume here that I really need new pens. Where the fuck have they all gone? I love how unprofessional these whiteboard streams are. My apologies, guys. So let's just assume here that these three here are introns. They would be removed and the extrons would be fused together to give you a complete molecule. Because remember, introns are non-coding DNA. So, from there we now have our mRNA. Cool. This was called pre-mRNA because it hasn't left the nucleus. mRNA leaves the nuclei. And this is where we move on to translation, which I would argue is quite easy. Kind of. It's weird. Okay, so we have the tRNA structure, which kind of just looks like a big-ass plus sign. But there are two main components you really need to know. And I'm going to draw them on red, because red represents, holy shit, danger. You got the amino acid binding site. And you got the anticodons. So, each tRNA is specific to one amino acid. I don't know any amino acid names. Actually, I know one. But I don't know any amino acids, so let's just call this amino acid OK. Bear with me, guys, this will make sense. So, here we have the, what do you call it? The ribosome. Imagine it like a burger. Again, this will make sense. I know my drawings are shit, but guys, this will make sense. So, we have our RNA in here. And what is going to happen is there are kind of two slots in the ribosome. So what will happen is these first three bases here, are called a codon. So what will happen is the first three bases will come and they will obviously line up here. Then a tRNA molecule, which is complementary to the codon, so it has, in other words, a complementary anticodon, will attach. And it will bring with it a specific amino acid. So, if these three bases, if this codon codes for the OK amino acid, then the OK tRNA molecule will bind its anticodon to that codon, and it will bring the amino acid with it. Now this process will repeat until eventually you have a big ass chain of amino acids. And from there we know all about the structure of proteins. We know that obviously it will fold or coil into a beta pleated sheet or an alpha helix. Hydrogen bonds will form, giving it a 3D tertiary structure. Ionic bonds will form, disulfide bridges will form, shit like that. And then eventually it'll be joined together through, I'm pretty sure, the disulfide bridges. I think they bond together the quaternary structure. So I hope that makes sense, guys. I really do. Because that, to me, has been the most difficult topic in biology. And if I've been able to kind of half-ass explain it to you guys in 10 minutes, I'm sure you guys can go out on your own and spend a couple of minutes researching this too. There might be some shit I've missed, but in other words, the anticodons are complementary to the codons, so only the complementary tRNA with the complementary amino acid will bring it to make the chain. So there you go. That was transcription and translation. I know Alex said that he hated the topic. I completely agree. It's a shit topic. But if I'm able to understand it, I'm sure that you guys will have an easier time understanding it too. So, we've done carbohydrates, we've done a bit of proteins, enzymes. Should we do enzymes, or do we think that won't appear? Fuck it, we'll do some enzymes. You got the induced fit model. The way this works is, and you have to be very specific in an exam here, you have to say that the enzyme, well, the active site, my apologies, the active site changes shape slightly. It slightly changes shape in order to be complementary to the substrate. That's all you need to know. Now, some little trivia, because I'm a sweat at this topic, I say that, I'm not really, but I revise like any normal person would. Because I know this topic quite well, I know that there are some keywords that you can put in your exam to guarantee you get marks. Those words are, you gotta put enzyme substrate complex. They fucking love that word, and if you don't know what it means, it's basically when the active site of the enzyme binds to the complementary substrate. That's another word, complementary. Talking about it being complementary, and this works for a lot of stuff in biology. The complementary tRNA molecule, the complementary antibodies, it's just bread and butter. That's your bread and butter. I'm trying to think what else. I guess just substrates, things like that. Here's a thing. Enzyme inhibition. Now, I only got 3 out of 4 marks for this in the exam, so I must have missed a point somewhere. But from what I can understand, let's assume that this big old bastard is the, the enzyme, okay? This right here would be the inhibition site. It's a part of the enzyme that is not the active site. It's a site on the enzyme that's not the active site. This is the active site, this is the inhibition site. Non-competitive inhibitors will bind to this site because they have a complementary shape. And what will happen is the inhibition site will cause a change to the active site structure. Now, I don't know how that happens. You could waffle about it affecting the tertiary structure of a protein. Maybe you would get a mark. I don't know. But that's non-competitive inhibition. Competitive inhibition is a whole lot easier. This inhibitor has a similar shape, a similar complementary shape, to the active site. It binds to it, but it does not form an ES complex. As a result, this enzyme is now saturated, and as a result, no ES complexes can form, meaning this enzyme is fucking useless. There you go. That's enzymes in like 2 minutes. Hope that helped. Very quickly, the changes in factors there are 4 that affect the rate of enzyme activity. You got temperature, so what will happen is when it reaches the optimum temperature, that is, it's basically maximum effectiveness before it begins to denature. I'll talk about denaturing in just a sec. Same with the pH level, and again, I will talk about why that is in just a sec. The changing concentration of substrates and enzymes, let's talk about this. If you increase, so let's say we have 10 enzymes. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Let's say we have 10 enzymes, okay? Let's say we have 2 substrates. Okay, cool. So what's going to happen there is that obviously 2 of the enzymes, 2 of the substrates will form ES complexes, la-di-da, the reactions happened. But if we added more substrates, more enzymes would then be used, more ES complexes would form, and as a result, the reaction happens faster. But what if we increased the number a lot more? Let's say we had thousands of substrates. Well, hold on a second, but we only have 10 enzymes. That's when the graph plateaus. That's when it flattens. Because all the active sites are saturated. And vice versa. If we increase the amount of enzymes, it would affect if you had dozens and dozens of substrates, yeah, increasing the amount of enzymes would work. Until that number surpasses the amount of substrates. In which point, you just have a lot of unsaturated enzymes that can't do anything. The graph plateaus. Denaturing. Denaturing is actually quite a specific thing. So, in case you guys don't know, you have ionic bonds between the positive and negative charges of a protein. In the tertiary structure of a protein, positive and negative charges. Now, these ionic bonds hold together the tertiary structure, but they can easily be broken by changes in pH. Obviously, a change in pH would drastically affect, it would break the ionic bond. If that happens all over the protein, you've completely changed its structure by denaturing it. And obviously, denaturing just means that the active site of an enzyme is no longer complementary due to the severe change of the tertiary structure. Now, we got the disulfide bridges between the cysteine amino acid. Now, the cysteine amino acid forms the disulfide bridges, and these are easily affected by changes in temperature, which will break them. And because disulfide bridges, I believe, don't quote me on this, because they hold together the quaternary structure of a protein, if you were to break a bunch of them, you would also break the quaternary structure of a protein, thus denaturing it. Hydrogen bonds are also easily broken, so keep that in mind. Speaking of, should we do water very quickly? So you got H2O. The O is negative, the H's are positive. This gives it a slight positive charge, but the main thing we need to know is if we have HOH, it means that the positive hydrogen is going to bond with other oxygen. Like so. So what this basically means is that you have bonds, hydrogen bonds. But these hydrogen bonds are weak as shit. So what that means is that they're going to constantly break and reassemble. Now, what does this mean? What is all this waffle about? What is bro yapping about? What it means is that it has some very unique properties. So many that I forgot most of them. So let me list couple that I can remember. Number one, they act as a solvent. What this means is that other, basically, polar molecules are able to dissolve in the solvents. Ionic polar molecules can form or can dissolve in this. And I don't really recall what that would do. I think it just allows it to form other ionic compounds. Next, great, Sammy's just spamming stickers. We're good, okay. And next, it has a high specific heat capacity. In other words, it acts almost like a buffer solution. In other words, it takes more energy, I believe 4.2 kilojoules, to heat up a kilogram of water. Meaning, again, it acts as a buffer solution. It retains its temperature. It's very difficult to heat up. Next, it is adhesive. So, the unique bonding between water molecules allows for adhesion. In other words, the molecules stick together. But the main one you can put is that it's a metabolite. So what the metabolite means, what weird-ass word means, is literally that you can do your hydrolysis and condensation reactions in it. Because, I don't know why, but it is. It's a metabolite. Metabolite? I don't know how you pronounce it, but that's what it does. Water's arguably one of my weakest areas, so don't get me wrong, guys. Speaking of which, gonna go take a swig of water now. Right, what we'll do 10 more minutes of revision, then we'll go do some exam questions, and then I'm going to fucking bed. Yeah, I love how, for context, I this isn't how I normally behave. I don't just randomly swear and shit, like I just did there. Basically, I just find doing these revision streams kind of funny. I know not many people watch them. In fact, I doubt anyone's even watching now. But I enjoy doing it. The thing is, I kind of suck at revision. I'm not the best at it. I know people have their little flashcards, mind maps. I don't like doing much of that. But I find I can do it incredibly well if I'm explaining it to somebody. Even if nobody's watching the stream, I still like to be able to explain it to an imaginary person. Genuinely, it's proven that that is a viable revision strategy, and it, in my case, helped really well. That's how I was able to pass a lot of subjects in GCSE. We had a really bad biology teacher, and literally the only reason I think I did half as well as I did is because I spent many streams talking to you guys directly about the subject. It doesn't help too that I've gotten really good RNG over the last couple of exams. I'm talking a lot of exam questions that I've revised have come up in the exam. Like word for word copies. That is incredible luck. I told Faith about it and she literally said, like, it's really lucky that that's happened so many times. And it is, because that won't happen in the actual paper. But I don't care. I'm going to keep doing it because it's revision. So, what have we done? Proteins, carbohydrates. I have not done codon tables ever. I don't understand them. So you know what we might do? We might spend a minute or so trying to do this in a little, because I've never done it. I was off on the lesson where we did it. What else could appear in the exam? Mitosis, but I've already gone through that. IPM, ATC. What else? Lipids. Should we do lipids? We'll do lipids. Lipids. So, lipids are joined together via ester bonds. But Joseph, how do these bonds form? Via a condensation reaction. Literally, copy and paste. You can use the word condensation reaction You get a bunch of marks. Oh, ATP. Remind me, we need to do it. I forgot to revise this. Ester bonds, so condensation reaction. Cool. You got the polar hydrophilic head, and you got the non-polar hydrophobic chains. These are fatty acids. That is glycerol. Glycerol backbone. That is a phosphate group. How cool. So that forms obviously a phospholipid bilayer. The way it does this is basically the polar hydrophilic head will want to point into the aqueous solution because it's water loving. These chains do not. They will face away from the water, and this forms a bilayer structure where the two sets of hydrophilic chains will point inwards away from the aqueous solution, and the two heads will point into the aqueous solution. Whether that's the cytoplasmic stuff or the extracellular stuff. I think that's all I really need to go through. ATP, adenosine triphosphate. It's made up of, you got adenine, next to, I believe, please correct me guys if I'm wrong, it's a, I forgot the word. Ribose sugar. 90% sure that's what it is. P. These are high energy covalent bonds. When they break, it releases a shit ton of energy. Cool. This phosphate here can basically be broken off to, again, release a shit ton of energy, but this then forms ADP. Basically, adenosine diphosphate. What this means, plus the inorganic phosphate. So, ATP, ADP, basically, when this P is released, it is then used to phosphorylize. It's then used to phosphorylize molecules. So, for example, the sodium-potassium pump is made for ATP. Basically, the P attaches to the sodium-potassium pump, causing it to change shapes so that it is now, and I forgot the word, but it is then kind of attractive to potassium molecules. And when the phosphate is removed, that's when it is then attracted to sodium molecules. So in other words, it phosphorylizes things, and then it's added back on in order to kind of repeat the cycle. I hope that that's everything. I hope that that is everything. Do you guys know anything else about ADP, ATP that I can't remember? Because I feel like it's relatively simple, actually. Is this a pentose or a ribose sugar? I can't remember, so let's just write it down. ATP structure. I saw an exam question where it was talking about what's the difference between, this isn't the start of some joke, what's the difference between ATP and DNA, of all things? And I thought, cool, I know this. I don't. Adenosine, DNA. Deoxyribose nucleic acid. Now, I struggled on this, so I want you guys to figure it out. I was torn between whether the sugar was different in DNA and ATP. But I'm an idiot because look at this. Deoxyribose, ribose. That's literally a difference. The structure of DNA is comprised of deoxyribose. ATP is made of ribose. It contains ribose. That would be a difference. A difference as well is that they contain phosphorus. Cool. These done. Another difference is that this contains adenine only. Meanwhile, DNA contains adenine, is it thymine, guanine, and cytosine? Something like that. ATCG. Obviously, an RNA would be AUCG. Remember, the difference between RNA is it's a smaller copy. DNA. We could very quickly do the structure of DNA fully. Prokaryotic and eukaryotic DNA. Let's do that. Prokaryotes and eukaryotes. Difference between prokaryotic and eukaryotic DNA. Prokaryotic is not bound in histone proteins. Eukaryotic is. Prokaryotic supercoils to be able to fit actually in the structure of a prokaryote. Meanwhile, eukaryote basically winds and then it, I guess, is compressed into chromosome structures. Shall we go quickly do some word things? Genes are a base code. It's a sequence of bases which codes for a polypeptide and a functional RNA. There you go. An allele is a different form of a gene. A locus is a specific point on a chromosome where a gene is located. You got the centromere, which joins together to homologous chromosomes. I might be wrong there. Chromosomes or chromatids. I'm honestly exhausted, and I've just spent 40 minutes going through a load of shit. So let's put it to the use, shall we? Let's go firstly check the structure of ATP. Actually, we don't need to. I know it. I'm going to go do some exam questions, but first, I want to do codon tables because I don't know what they mean. And if we get asked a 5-mark question tomorrow on codon tables, I will cry. So yeah, I know that these revision streams aren't like too popular. Like, you know, I was kind of hoping what would happen is I started to do more of these consistently. We would get more people in, and it would be a lot more interactive. As in, like, you know, if I made a mistake, somebody could comment and be like, "Oh, you made a mistake. This is a." You guys could comment with like exam answers. Thing is, I know how fucking boring that sounds, and sorry for the language, but you know what I mean. During the COVID days, I barely attended the online lessons. Well, I did, but I would always be playing a game in the background, or I would be doing some other shit. You know what I mean? I never enjoyed just sitting there listening to a teacher yap about a topic that I didn't understand. So, I know exactly how you guys feel when I ask you guys, hey, I'm doing a revision stream, do you want to come on? Fuck off. I bet that's what a lot of you are wanting to say. And I honestly don't blame you, because if I saw someone doing a stream for some shit that I didn't understand, I would basically say the same. Thing is I understand it, you guys might not, and I'm not that good at explaining things that other people might not understand. So, let's go do... So I don't think there'll be many questions in digestion and absorption. There might be some questions on co-transport, but I think I know that well. Genetics... Okay, here. And then we'll do some questions on maybe ATP. So, DNA, genes, and chromosomes. We'll look at the question paper, mark scheme, transcription and translation, question paper mark scheme. Okay. This question is about mitosis in cells. Name the fix... Oh, wait, this was a question on the exam. Okay, we're 40 minutes in. In case I send this to somebody later, I'm literally looking at a question which was on the exam. So this will definitely be helpful. This question is about mitosis in cells. And this is a cell in what looks like prophase. The chromosomes are all coiled together, basically condensed. Name the fixed position occupied by a gene on a DNA molecule for 1 mark. This is a locus. Basically, if you had a chromosome, it's just a bit where a gene is. Describe how a gene is a code for the product of a polypeptide. Do not include information about transcription or translation. And it's 3 marks. I'll be honest, when I read this in the exam, I sat there and went, what the hell? What is it asking for? If we're not allowed to talk about transcription or translation, I think this is more keyword stuff, right? Like, you need to basically say that a gene is code for a polypeptide. It's a base sequence code for a polypeptide and a functional RNA. But I don't know what else. Describe how a gene is code for the production. I guess you could say that... I guess you could say the gene's codons are complementary to the anticodons of tRNA, which brings a specific amino acid, forming a polypeptide chain. I don't know. I'm honestly lost with this question. What's it asking for? Wow, that was easy as hell. This is all waffle. This is all waffle. Okay, here's the answer. Because the base nucleotide sequence, so obviously nucleotide, I forgot the structure of it, like this, and then that's the base. So the nucleotide structure, the base sequence of the nucleotide is in triplets. Yeah, we knew that, codon. But this determines the sequence of amino acids. That's all you had to put. Basically, the structure of a nucleotide affects the, arrangement of bases, or affects the sequence of bases, which in turn affects the amino acids, the sequence of amino acids. So let's try to put this into tangible terms. The sequence of bases on the nucleotide is arranged in triplets, and these triplets code for a specific sequence of amino acids. And obviously amino acids are the primary structure of a protein. Okay. So that was a goofy little question there, but very badly worded. Describe the term exon. Ooh. Exons are the coding genes. So they are genes which code for a polypeptide. Or I guess bases which... non-coding DNA. Would I just put coding DNA there? Or what do you guys think? Define the term exon. Base sequence coding for polypeptide. Bruh, it's literally the answer to this question. It's a base sequence coding for the sequence of amino acids. It's a base triplet sequence coding for polypeptides. So in other words, what I said, but in way more detail. instead of coding DNA, it's a sequence of bases which code for a polypeptide. And that's what a gene is as well. A gene is a base sequence of DNA, a base sequence of triplets which code for a specific, The table shows mRNA codons for some amino acids. Figure 1 shows the DNA template. Use the information to give the amino acid sequence determined. So you got AGG. So it would be glycine, TGC, is not there, so that would be non-coding. CCT is non-coding, and GGA is glycine. So would it just be glycine? What the fuck? Serine, what the hell? How did that make sense? Wait. Serine, aline, glycine. You know what it is? It's a DNA template. I didn't read the question right. It's a DNA template. What that means is we need to convert it into RNA before using the table. Okay, got it. Very quickly then. AGG, CGT, CCT, and GGA. Okay, so AGG, so hold on. So it would be ATCG, so that would be AU. Apple's under. I hope I'm doing this right. So that would be UCC. That would be GCA. That would be GGA. And that would be CCU. Let's check. So, UCC is serine. So I'll just put ser. CGT is I must have done this wrong. Would T just be air? Sorry, I'm doing this one. I'm a fucking idiot. Sorry, GCA. Where's GCA? GCA is alanine. Then GGA is glycine. And CCU is pro. So Sir Alagloy Pro. Let's see if that's right. Sir Alagloy Pro. There we go. We did it. We did it right. Okay. That is kind of a deceptive question. You have to literally translate it into its RNA base sequence before being able to read it. For 1 mark. That took like 5 minutes, bruh. For 1 mark. Oh, shit. Describe how a phosphodiester bond is formed between 2 nucleotides. What the? Am I going to get clowned on here for saying, is it just a condensation reaction, or am I underthinking? I swear, condensation reactions is how every molecule is joined. Oh shit, people have been commenting. Hold up. I have $1 million idea. With my autism money, I can put into production, drop out if I wish. What do you mean? You are not dropping out of school because you get £95 a month. Bro, you need at least like 1K to survive. So it's this. That's a phosphodiester bond. They are formed when the enzyme DNA synthase acts on it. So I'm assuming that DNA synthase catalyzes the condensation reaction. And I don't know whether I would need to describe what condensation is or not. Let's check. Condensation. Yes, there we go. 3 marks. Condensation reaction between... Okay, so I would have had to specify between this and this, yeah. Condensation between the phosphate and deoxyribose catalyzed by DNA polymerase. There we go. That was easy. That was an easy 3-mark question. What the hell? The 2 DNA strands of a particular gene contain 168 guanine bases. The relationship between the number of guanine bases, so remember G binds with A, is a formula. So the number of G bases is 4. Whoa, is this an algebra question? What the fuck? G equals 4, A plus T minus C. So if G equals... Oh no, wait, this is a nice question. A plus T minus C. Okay, so hold on. Okay, no, I think I get this. I think I get this one. A little bit of math required. 168. So 168 is the equivalent to 4 A plus T minus C. So there must be 168 adenine bases. Right? AUTC... Oh no, I'm an idiot! Sorry, I'm so sorry, guys. No, I thought it was uracil for some reason. There we go. Okay, that makes a lot more sense. 168 is equivalent to something minus 168. So we have x minus 168, or basically 168 minus 168. So when we add 168 to there to get 336? Yep, 336. So we would add that there. So now we get 336 is 4A plus T. We would divide by 4. So A plus T is the same as... $48. So 84 is the same as A plus T. So what that means is there must be 42 adenine. So that means 42 plus 42 plus 168 plus 168, which I'm going to guess is in the ballpark of maybe 600. Okay, I got the right answer there. I got the right answer, 42. And it was in the ballpark of about... I said in the ballpark of 600. My estimation was way off. Yeah, 600 makes complete sense. There we go. I got an exam question right, and it was using algebra. Holy shit. Name the protein associated with DNA in a chromosome. That, my friends, is a histone. Sorry, imagine if it wasn't there. That would be so funny. Ready? Yes, histone. All right, we're rocking. We're rocking in the free world, guys. Call me Nardwar, the way I keep rocking in the free world. I don't know why I did that. That was so funny. Let's grab some of these super quickly. Rick Astley badge. I'm losing the plot, guys. We're about an hour in now. I am feeling happy. We have answered some questions very well there. So, what can we interpret from this? Number one, a extron is a base sequence of triplets which codes for a specific sequence of amino acids. That's all I've learned. Should we... I'm trying to think. Is there anything else I need to revise? You know what? It wouldn't hurt to go downstairs and check very quickly. Where am I for lesson, please? I'm making racks. What the fuck are you on about? I'll be a couple of minutes, go downstairs, grab my book, grab a drink, and I'll make sure that I've revised everything for this exam. Catch you guys in a sec. Here we go. Let's go check what we need to revise. A genome is... What's a genome? The entire set of genes in an organism. Proteome is... Speak. What happened? Hello. What do you mean, what happened? No, so seriously, a proteome is the set of proteins produced by a cell. That's it. Oh, here. Okay, ready? We got transcription and translation, which we've just gone over. Like, look back on... Okay, 1 sec, my guy. What are you... Bro, this is going to be an awful troll. You're going to be like, oh my god, look at 3 minutes 52. Monosaccharides and poly, we've just done. Amino acids, we've done. Yeah. Polypeptides, we've done. DNA, RNA, tRNA, we've just done the structures of. Chromosomes, we haven't done, so we'll go do that. Enzymes, let's go! That's everything. I have unintentionally just revised everything that is in the paper by accident. That's everything that's in the paper. I guess it wouldn't hurt to go quickly look at the chromosome structure, which we're doing now. I've literally predicted the whole paper here. Am I good at what? No, I'm okay. Let me look back at the stream. What are you waffling about? You died. Yeah, I don't know about that, man. Goddamn, conversations with people. Crazy how one day the legends forget the equal on this under pressure sequel. We're not taking it back. We keeping it pushing. I can't. I'm using my phone to record. Okay, you said like a couple of minutes back. What the fuck? Hold on. It just goes dark. I just walk off and it goes completely dark. Have I just a real... Okay, no, I'm taking my piss. Oh wait, before I went away. What do you mean before I went away? I was playing into it there. I was trying to troll you by, I just turned off the light. That's all I did. Look, I just did that. Before, what happened, like, actually? Okay. 1 sec. Because my MacBook is shit. Because it's a piece of shit. Goddamn. Conversations with people. Crazy how one day the legends forget that they equal on this under pressure sequel. We not taking it back, we keeping it pushing. Deep in the cushion. Guys, I'm a Logic stan. I'll be honest with you guys. It's downloading. You know what we'll do? We'll do another question, and then by the time it's loaded, it should be done. Because it's taken a while. Sorry about that. Okay, DNA in the nucleus of a plant cell compared to prokaryotic. Ooh, tricky question. Scientists investigated the genetic diversity between several species. They studied the non-coding multiple repeats of base sequences, define non-coding base sequences, and describe when the non-coding multiple repeats are positioned in the genome. Oh shit, I don't know that. Non-coding base sequences are introns, which means that they are removed during splicing before the pre-mRNA becomes RNA. I don't know where they're found, though. Are they just found at the ends? DNA that does not code... Okay, way easier definition here. DNA that does not code for protein/polypeptides. And it's positioned between the... Oh, yeah, well, no, obviously I knew that. It's just positioned in between genes. Okay. So we now know what an intron is, too. An intron is a sequence of bases which does not code for a polypeptide chain. Right, what did you send? Or is this something really bad? What's this going to be? That's all I've won. Should we... I'm trying to think. Is there anything else I need to revise? You know what? It wouldn't hurt to go downstairs and check very quickly. Where am I for lesson, please? I'm making racks. What the fuck are you on about? Oh, what the hell? That was not me. That was unironically not me. What I did is here. Let me try recreate it. Did something similar happen there? Did something similar happen there? Because I think that's just when I scroll up to check notifications. Yeah, there we go. You don't want to meet my dark side, Batman. You don't want to see my dark side. Okay. Anyways, let's keep it going. Let's keep it going, guys. So, introns are just base sequence of a base sequence that does not code for a polypeptide. And they are found in between the genes, which makes sense. You'd want to kind of buffer them out. Yeah, I like that. The nucleus and a chloroplast of both a plant cell both contain DNA. Give 3 ways in which the DNA of a chloroplast is... We've done this question too! This was in the exam which we did. Holy shit. 3 ways they're different. Okay. In a chloroplast, it's circular DNA. In a nucleus, it's chromosomal linear DNA. 2, I'm going to say that the chloroplast DNA is smaller, so it's shorter than a longer. And 3, I'm going to say that they're not wound in histones. Let's go. DNA is shorter, fewer genes, I didn't put that. Circular, let's go. Not associated with proteins and histones, let's go. 3 marks, easy shit. Some DNA nucleotides have the organic base thymine, but RNA nucleotides have uracil. Give one difference between DNA and RNA. Bro, these questions are easy as shit. You could put so much here. You could say RNA is just comparatively shorter. RNA is obviously a single strand of nucleotide compared to the double strand. Holy shit. tRNA molecule. I've done this question too. Name the structure W and X. X is the anticodon. W is the amino acid binding site. There we go. Not all mutations cause a sequence of a gene change. I don't know why this would be. Logical. What if the mutation occurs in non-coding sequences, i.e. introns? And what if it, obviously, because a triplet codes for an amino acid, if one of those triplets is affected, then if 1/3 of the triplet is not affected, it's obviously not going to affect at all. Cool. Wow, okay, unironically, that's it. All the other questions we've seen so far different in histones. Yeah, that makes sense. All of the rest of these questions are either comparing DNA and RNA or are comparing something. Finally, name the type of bonds between the base pairs hydrogen, nucleotides, phosphodiester. 2 differences between tRNA and mRNA. Bro! Okay, hold on. tRNA has anticodons, RNA has codons. tRNA has a... Oh, I don't know, actually. tRNA... Oh, yeah, I was actually going to say that. tRNA is a clover leaf shape, mRNA linear. tRNA has hydrogen bonds. Didn't know... Oh, yeah, I did know that. tRNA has amino acid binding site. Yes, obviously, guys, state the obvious. It works. In a eukaryotic cell, the structure of mRNA used in translation is different. Explain the difference between the mRNA molecules. I wouldn't know. In a eukaryotic cell, the structure of the mRNA is different. Oh, wait, different to pre-mRNA. Oh, well, I know that one. Let me bring you guys down here. The difference is, pre-mRNA, you got, obviously, the introns are still there. The non-coding DNA is still there. It is not being spliced, and that means that the extrons are not joined together. 2 marks, that should be a good definition. So yeah, mRNA has fewer nucleotides. mRNA has no introns. Okay, and I would have had to specify because of splicing. Describe how one amino acid is added to the polypeptide. So this is a 3 marker. So it joins to the ribosome. So hold on. The tRNA molecule with the complementary anticodon to the codon in the ribosome joins to the ribosome and brings with it an amino acid. So the tRNA brings a specific amino acid to the ribosome. Oh, I didn't know this. Hold on, this is new info. The anticodon binds to the codon. I knew that. Amino acids join by condensation using ATP. Okay, condensation uses ATP to join the clover molecule so cool. I genuinely did not know that. That's a cool little statement. I was kind of going a bit off the rail there, talking about stuff that wasn't related. Final question. Describe how mRNA is formed by transcription for 5 marks in eukaryotes. Okay. So, I'm fucking tired, guys. I'm gonna be real. I'm so close to ending the stream. Let's just check. Let's just very quickly. So, it unwinds due to the hydrogen bonds breaking, thanks to DNA helicase. You got, obviously, the free-floating RNA, phosphodiester bonds. Let's just check the mark scheme. I'm so tired. Hydrogen bonds break. You can't say DNA helicase. Okay, that makes sense. So, but hydrogen bonds break. One strand of DNA acts as a template. The free-floating RNA molecules align to their complementary bases. RNA polymerase joins the nucleotides. So in other words, it joins the phosphodiester bonds together. And then pre-mRNA is spliced. Cool. So the only bit we missed there is that it's only one base. Only one nucleotide strand is used in the process. And we missed just 1 or 2 minor words. Honestly, I am so tired now. And it's not even 8 o'clock. I'm so tired. I'm asleep now. Because the exam is first thing in the morning. So, thank you guys for watching. I appreciate it. I will be off now. Hopefully I'll remember some stuff. In fact, you know what I might do? Just while I'm, still half alive. Where's my pen? What I'll do, I'll go write some notes on the board. Just for... I mean, most of it will be to do with genes, and if a paper isn't about genes, this is going to be completely useless. But you know what? So obviously we now know that when the anticodons and the codons form, they link together, and this causes the amino acids to join via condensation. Which makes sense, yeah, but I wouldn't have realised that it used ATP. We know introns are just so basically a base sequence which does not code for polypeptide. You knew that, great. Bro did not know that. Obviously, extrons are a base sequence which codes for a sequence of amino acids. I don't think there's much else to put. So you know what? I'm wrapping up the stream. Guys, unironically there, I just sat through 1 hour and 15 of doing revision for you guys. So I hope any of you found this useful. If you did, be sure to, like the stream, share it with your mum. Don't do that, because I don't know. So yeah, I'm very tired, so I'm heading off. But genuinely, that has been good. For those who are just tuning in now, we literally just revised every topic which our biology teacher suggested we revise a couple of days ago. She literally went through the paper and recounted every single topic that would appear in it. If that isn't good revision, I don't know what is, because I literally just went through every topic that will be in the exam tomorrow. Now, I'm gonna be real, I probably won't get 100%, obviously. There's going to be some really tough questions, I can imagine. I honestly always say, anything above 50 is good, right? Think about it this way. You need about 70% for an A, I believe. 75 for an A star. Getting 60% is like a B. Getting 70%, that's an A. So anything above, that 50 threshold is good. If you can guarantee at least half marks, then you're doing something right. That's the way I see it, at least. And yeah, obviously, I hope you guys did enjoy. Again, go rewind the stream, because after I end the stream, it becomes really difficult to watch. It kind of becomes really choppy for a couple of hours. So go rewind the stream now. I talk about carbohydrates right at the start. I talk about ATP, ADP, nucleotides, DNA, RNA. I talk about translation, transcription. Go watch it. Goodbye.